When you're talking about biocompatibility of medical devices, chemistry is a part of what you need to take in consideration to test your device. Basically, you need to make sure that your device is safe also from a chemistry point of view. If you have a catheter that you want to use to treat a patient, to administer a drug or whatever, that catheter, it needs to be safe. It can't be that you're using a drug to treat your patient and that your patient becomes ill because of the catheter you have used. So you don't want that. So the biological safety is important, chemical safety is important as well. We have an Iso10993 chapter, so chapter 18, that is covering also the chemical part of the medical devices. When you are talking about chemical characterizations of medical devices, some natural responses of people that are new to it is like this. They are frightened about it, say, what the hell is this? Why do we need to spend such an amount of money on a device? We know it's safe. Yeah, they might know it's safe, but it's still an important test. You really need to include your chemical evaluation of your medical device. The norm, it has been renewed recently. There's some different phrasing, definitions, and so on. There is also the concept of the AT, of which I will talk about later. But most importantly, and I can't stress it enough, in orange, you see there is a first step before you go into the lab and you're doing extractions with solvents and using fancy machines that cost like millions of euros or dollars. It's a paper exercise. Whatever you need is information from your suppliers. You need your brain, you need a paper and a pen, maybe a laptop if you're fancy technology. Well, and that's so important that you do that technical or that theoretical exercise, because you think about your materials that you're using. How are those materials processed? What kinds of chemicals are we using in the processing? How is it stored? What kind of manufacturing aids are we using? That's the basics of chemical characterization. You really need to think about what is our device, how are we making it. You can write it up in a document. That's the first step in assessing the risk of the chemical safety of your device. That's the first step. Only after you've done such an assessment, it can be that there is a conclusion, you have to do some chemical testing. But don't forget that first part in gathering information. Your suppliers, the suppliers of your suppliers, the entire supply chain can help you out on this. If you don't have the capacities, ask experts to assist you in this process, because it is information. It's critical information. Only after that, you go into the lab, you do your extractions if it's really needed. When we are talking about an extractable study, it means that we are using laboratory extraction conditions and vehicles. It's worst case, it's organic solvents. No, it's not vegetable oil. But it's chemistry that we are doing, so it's not biology. We use alcohols, we use hexane, for instance, and we are really putting lots and lots of stress on the devices from a chemical point of view. When we talk about a legible study, we are looking at real patient contact. 37 degrees, most probably. Maybe we include a fever, 40 degrees C. We look at the length of a therapy, of a treatment of a patient, and then we really try to simulate the real clinical exposure to a patient. So when we are talking into metaphors, if you have a piece of rope in an extractables study, the rope is so tense, so stressed, it's almost breaking. In a legible study, we don't apply real stress. It's just normal circumstances as is important for a patient. When we go into an extractable study, there are a couple of parameters that you need to cover in designing a study. 6, but we added number 0 as well, the test item, what we call the device itself. There are parts of a design that may not be relevant for chemistry testing. If you have an infusion set, a piece of tubing through which a saline solution or a drug product is flowing, many times there's the plastic clip to clamp the tubing to stop the flow. Don't include it in your extractable studies, because the only way it's contacting the patient or the healthcare professional is by touching with the fingers. Like every day you're touching your computer mouse, your pen, you're doing the cooking in your kitchen. So it doesn't make sense to include such parts of your device. Solvents, I mentioned we are doing chemistry. We don't use vegetable oil. We don't use media in which cells are happy to grow. We are using solvents. Regulatory authorities, they ask for polar and apolar solvent, just to bracket the entire spectrum of the polarity of. And we use water. It's a polar solvent. Most cases, as an apolar solvent, we choose hexane. Intermediate polarity is often requested as well, especially for permanent implants. The most likely candidate for a semipolar solvent, that's isopropanol. We need to consider the use of the device. And that is the part on which I will elaborate much on at the end of the presentation with the case studies. But if you're having an infusion bag with saline, it doesn't make sense to do an extraction with hexane. Hexane chemistry data will not tell you anything about what is happening in a patient or what will happen. So, at least the bag, for that bag. So, really think it through, and I'll talk about that later as well. But using hexane on other types of polymers, hexane is the best apolar solvent. But yeah, what is happening is, for instance, this is a piece of tubing that we cut in smaller pieces just for the visualization of it. If we do an extraction with isopropanol, we see that the isopropanol, after an incubation, it turns yellowish, greenish. It's not okay. When we do the incubation in hexane and as an extraction solvent, it's really swelling tremendously. What is happening with isopropanol or hexane when it's in contact with these pieces of tubing, it's not compatible. The solvents, they are chewing up your molecular backbones of your polymer. Solvent compatibility with certain devices, with certain materials, is an issue. It's something that we need to be careful for. And that is also what is in scope of investigations and trying and saying what makes sense for our device, for the patients, and so on. So that is also where one protocol fits all is not always applicable. We need to apply extraction conditions. For instance, the extraction ratio, basically we have one device, how many milliliters of solvent are we adding to a device to come up with, or to make the extraction itself. We have this table from the Iso10993 standards. It's easy. Well, in theory it's easy. In practice, it's not always easy, because when you're doing the extractions of small screws, or you want to do an extraction of an entire hip or an entire leg, it's completely different sizes, shapes of extraction vessels that you need to use. And it needs to fit in it. You want to maintain the extraction ratio that you put forward or that you desire. But yeah, sometimes the real world does not allow you to do so. Creativity is really key, and sometimes, having a deviation of what your desired extraction ratio, it happens. Extraction times and temperatures, again, nice guidance. Either it's an exhaustive extraction condition. Basically, in an exhaustive extraction conditions, you prove that you have extracted almost everything out of your device as is possible. Or you can go for exaggerated conditions. Basically, you do a 72-hour incubation at a 72-hour incubation at 50 degrees C. You can do with a lower temperature or a shorter time. For instance, simulated use conditions. You can do it if you know that your device has a limited contact device. For instance, it's only used, your device is used for 10 minutes during surgery, only once in a lifetime. Obviously, then you can write a justification to use a simulated use condition. Once we have generated the extracts, we do the analytics. That's where the fancy machines come in place. We analyze for the small molecules. Yeah, it's represented by a child. It's a small molecule. When we are talking science, we are say the volatile organic compounds. We also analyze for the medium-sized molecules. Yeah, with the normal adult picture. These are the medium-sized molecules. These are the semi-volatile organic compounds. We also analyze the big molecules, non-volatile organic compounds, and metals and some other compounds that are maybe of interest. It's a big scope. We want to, let's say, we want to screen for our extractables, and that's kind of symbolized with the metaphor of net fishing. You throw a net in the ocean or in the sea or in the lake. You want to try to catch every fish within your range. You're not interested in only one specific piece or species of fish. No, you're interested in everything, and that is screening. The methods, the headspace GCMS, the GCMS, the LCMS, they are optimized. The detector range is as much open as possible to detect as many chemical compounds as possible. That's different, of course, for metals. I mean, there's not a zillion types of metals or elements in our universe. That's a limited set. There is guidance on which elements and metals you should test for. That is a targeted technique. Targeted, yeah, I'm not really a fan of hunting, but it's the metaphor that we use often is hunting. You are interested to hunt a deer. If there's a rabbit, you will not shoot it. You're hunting for the deer. And same in analytics. You are interested in cadmium, mercury. You're analyzing those metals, for instance. Sensitivity of the analytics, it's also important. For the people that are not into chemistry, this is a chromatogram, the output of a fancy machine. Every peak that you can see, a peak is, let's say, a Gaussian curve. Most preferably, it's a very narrow Gaussian curve. Every peak is representative for one extractable, just one type of molecule. The higher the peak is, that's the measure of how high the concentration of that chemical is in your extract. And now, the big question is, if you have such an output of a machine, how many of those peaks should we report towards our customers? Only the highest one, the two highest, 3, 4? Or should we go really to the bottom and saying, yeah, maybe if you zoom in, you find 30 small peaks as well? That is where the concept of the AET, and that's the abbreviation of analytical evaluation threshold, comes in place. That threshold, and then the definition is the threshold below which the analyst, so analyst is the person working in the lab, needs not to identify or quantify the legible or extractables or report them for potential toxicological assessment. So basically that AET concept, the threshold, is a horizontal line, and how high or how low you place that horizontal line on the chromatogram is whatever peaks that are above it should be reported or not. If a peak is below that threshold, there's no concern, there's no toxicological concern, and you can just ignore those peaks because they're too small, concentration is too low, and we all know the basic law of toxicology: the dose makes the poison. If even a toxic substance is present at very low concentration, it can be that it's not poisonous, basically. That's what this line is intending to say. Of course, we don't choose that threshold, it's not at random. There is some mathematics behind it. The norm also helps you very well. It's based on how is the patient exposed to the device, short term, long term. How are we doing the extractions? One device in a liter or one device in 1 milliliter of extraction solvent? How are we treating our samples prior to the analysis on the instruments? What is the quality and the reproducibility and the uncertainty of the analytical chemistry of the methods of those instruments. These are all factors, they are summed up on the slide, that we take in account, and if you have values for all the parameters, you come up with a value, an analytical value, so it's expressed in micrograms of a chemical substance per milliliter of your extraction solvent, of your final extract. That is how we, as an analytical lab, we can place that horizontal line on the chromatogram. It's either high or low, or very low, or extremely low. Whatever we have to make sure is that the sensitivity of our analytical method is appropriate for your device, for your AT. Basically, the limit of quantitation, the limit of detection, indicated with the green and the red line on that chromatogram, your AET line should always be higher than your limit of quantification and your limit of detection. And that is our challenge. We need to make sure, we need to be creative on how we deal with this. We can push this to the extreme. We are a good lab. Although I'm wearing glasses, I'm not Harry Potter. I don't have the scar on my head. Science, chemistry, it has limits. Also there, once you're hitting those limits, it's key to start talking to your chemistry lab, to your toxicologist, to device experts, biocompatibility experts, to find a way out. A little bit like an escape room. And there are always solutions, right? There are other compounds. Cohorts of Concern is the name. Specific chemical classes that are particularly a concern for patient safety. You could go into testing each compound that is on such lists, on cohorts of concern, but in most cases this can also be a theoretical exercise. If you know your materials, you have control on your production process, you have the knowledge, you have the chemist on board, you can make a theoretical exercise to justify and to predict that there will not be such chemicals from a cohort of concern present. Identification is also important. Identifying your extractables is key because the identity of your chemical, and if your identity is correct, then a toxicologist can really work. If the toxicologist has no identity or an incorrect identity, whatever you will produce or whatever your toxicologist will have as an assessment, it will be doubtful. So having good identifications is key. Confirmed identifications is what you need. Every peak in the chromatogram has a recorded mass spectrum. A mass spectrum is basically the fingerprint of a molecule based on its mass of the molecule and its fragments. Because when we are measuring the mass of a molecule, we are also destroying the molecule, and we measure the fragments as well. That is what you see on the left picture. And we match that mass spectrum with our library. At Nelson Labs, we have our in-house library. It's a big library. We can do that matching, and we come up with an identification. There are also commercial libraries, like the top suggestion is saying, but it's never been designed for extractable or leachable compounds. It's a huge library with many mass spectra in it, but it was never designed for extractables or leachable identifications. So our in-house library at Nelson Labs, and we are actually very proud of it. It's about 6,000 compounds. We started building that database 20 years ago, and we are still building on it. It's a never-ending story because, med device industry, you are very innovative. You come up with new materials, new production methods, and so on. And that means that also we as a service provider, we continuously invest in our lab to try to identify all those extractable compounds coming from your devices. And having those reliable identifications is the key for a toxicologist to do a meaningful job. Quantification, yeah, I can talk an entire day on the quantification of extractables, but in short, when we are doing a screening approach, we are really estimating we are doing semi-quantification of our extractables. It's not a full quantitative result, but the estimation is good enough for a toxicologist to work with. In a targeted approach, when you know which molecules that you're hunting for, you have mostly optimized methods, many cases validated methods. You can really work towards really a full quantification with known accuracy, with known precision, and so on. But that's only when you have defined a specific set of molecules that you're interested in. When you're doing an extractable study, you're interested in everything. Everything you see, you want to, let's say, at least have a good estimation of the quantity. And that is the key. So the two bottom bullet points are what we are doing in an extractable study. For metals, we have a full quantitative method, for instance, because we know which metals we are hunting for, so we have optimized our method and validated our method. And then we make a report. And to give you an impression, a report, yeah, it has lots of content, and then there's lots of tables with, for instance, chemicals, with their unique identifier, how confident we are in the identification, and also what is their concentration, how many micrograms of a chemical have we found to be extracted from one device, right? Then we go to the toxicologist, or you go to your toxicologist, and the toxicologist does the assessment. Yeah, if you're lucky, all the extractables are found to be safe at the concentration level they were extracted, okay? But sometimes there are compounds that cannot be considered as being safe because their concentration is too high, meaning the margin of safety, MOS, is smaller than 1. It's saying the extracted amount of the extractable is too high, and if this entire concentration of that extractables is being exposed to a patient in the blood and fat tissue, in the bone, on the skin, whatever, then there is a risk for instance, the development of a cancer. So that is not what you like, obviously. But then, yeah, a legible study comes in place, because in a legible study, you are not putting that much stress on your device. You are using much more mild conditions that are in line with the patient exposure. And for instance, this was a case, there were 2 extractables present at a very high concentration, an aliphatic chain, nonane, and DEHP. It's a phthalate. It's a very frequently used plasticizer, in, for instance, DEHP, to make it soft and flexible. After a legible study, we found that the amount of DEHP that spontaneously migrated from the device to the contact medium was low enough to be considered as safe. But that's only because the conditions were much more mild. In the extractable study, it had huge concentration because you were doing your extractions with isopropanol, with hexane, and that was not safe. In a legible study, the same chemical entity became safe. You were able to prove it. And then in the end, your toxicologist can also acknowledge that the risk, the safety of your device is acceptable. Now, some case studies. If we have a simulation study of a device with indirect patient exposure, and the example here is a plastic bag that contains saline for infusion. In many cases, we get questions and we say, well, we know it's a device because there's no active ingredient in there and so on. But we still refer to the pharmaceuticals guidelines, because what the pharmaceutical guideline says, yeah, use your or do your extractions with relevant solvents. You don't do an extraction with hexane if your bag is only in contact with the saline solution. Then, instead of using a 72-hour incubation at 50 degrees C, well, look at the shelf life of your bag. Is it 1 year, 2 year, 3 year? And work towards that shelf life. It's much more meaningful than doing a brainless test for 72 hours at 50 degrees C. And important is also that you test your legibles in this case. We can talk about legibles because we are simulating the use. We can do multiple time points within a shelf life. For instance, you have freshly produced bags, time point 0, after 3 months, 6 months, 12 months, 18 months. We take a sample, we analyze the saline solution, and we can analyze for leachables. Saline solution is a very good solution because also from an analytical point of view, that's a clean solution. It doesn't interfere, it doesn't introduce any noise in our analysis. So this is very, very, very meaningful. And in the end, we also want to see the trend of your leachables. Most cases, they go up. Top left example is that's a linear release of your leachable compounds into your saline solution. But yeah, bottom right, it can also be that there are leachables, most particularly if your bag is semi-permeable. It can be that the highest concentration of a chemical, of a leachable, is not at the end of the shelf life, but that you're hitting, you're going down again because of evaporation through the layers of your bag. Another case study, it's again bags and tubings, but it's, let's say, an enteral feeding set. So you have the bags and then there can be administration to the stomach or to the gut, depending on the type of treatment of the patient. We don't need to test the full shelf life because these are devices and they are filled with a feeding solution or a hydration solution just before the actual use. And it can be used for a certain of time. And yeah, you have the repeated use of a device, and it continues for days, weeks, months, who knows. All right, step 1, eliminate all the irrelevant parts, so the plastic clamps, everything that is not in direct contact with the feeding solution or the hydration solution, or remove it. You need to use a wide coverage of solvents because, it can be used for hydration, aqueous solution. It can be used for, to feed proteins, but it also has lipids in it because, you need to have a balanced diet. Even if it doesn't go through the mouth, you need to have a balanced diet. And you use exaggerated conditions, for instance. Basically what you're doing, I'm sorry for the amateuristic pictures, but that's what you're doing. You're putting it in an oven. Really simple as that. You end up with lots of extractables. Why? Well, solvent compatibility of all your materials. If you're using an EVA bag or a polypropylene bag or whatever, all those polyolefins in combination with hexane as an extraction solvent, they are basically not compatible solvents. They are too aggressive. Exaggerated solvent exposure, you are extracting maybe inside and outside. You are extracting parts that are maybe the top of the bag is not in contact with solution, or only a few droplets are in contact with the opening at the top of the feeding bag, and the most of contact is only at the bottom of the bag. You're having a huge overrepresentation of certain parts of the system in your extractables data. Irrelevant extraction conditions, 50 degrees C. Why? It's such a hot temperature. Basically, what we're feeling, or that our customers can be feeling, to be that donkey, and you are trying to work with a huge load of extractables data. yeah, the picture says for me much more than trying to explain the feelings of ourselves, of our customers when dealing with such data sets. And we want to try to find a solution for that. And yeah, it's in a simulation study. What is the use of such feeding bags? Yeah, room temperature, yes. Maybe 37 degrees C for, yeah, if children are in the population and the feeding solution needs to be heated a little bit. But yeah, 37 to 40 degrees C can be the maximum, not 50. 24 hours is the maximum time because, for instance, the IFU says replace or use every day a different bag. Find suitable solvents. People are not fed with pure vegetable oil when they are using an enteral feeding set. So why are we using hexane as an apolar solvent? We need to find better simulating solvents than hexane. And what is also important is investigate the kinetics of the leaching process. How fast is it going? Is the release, are the chemicals released after a 5-minute contact? Or is it a real slow release? So that also a toxicologist knows we did 24-hour simulation extractions, but the real use of the bag is maybe 12 hours or 8 hours. Then there is still a scientifically proven release kinetics and saying, look, we have the 24-hour data, but we can make a deduction and saying what is the exposure of a patient after 8 hours or after 12 hours, which again can help to mitigate the risk of being exposed, or it can help mitigate the risk of making an incorrect judgment by the toxicology by having better data. The regulatory authorities, they are not experts in your device. The person that reads your file, they are not experts in the processing, the production of your materials. They are not experts in whatever you're doing. At least that is what you have to assume when you're writing your file. Basically what you have to do is you do storytelling. You take them on a journey and saying, look, yeah, it's childish, but the reviewers at the regulatory side, they need to understand why you chose 24 hours. If you say, yeah, we chose 24 hours, and you stop, no. You say, we chose 24 hours because we know the maximum exposure of a patient to a bag is 24 hours. Every day a new device is used, and in most cases, the treatment stops already after 8 hours. That's the story that you tell. And once they get the story, they will also understand why you are making certain judgments and decisions. Basically, if you're going from an exaggerated study, you have maybe 200 or 500 extractables compounds. I mean, people that are selling tox assessments, they might love it because it could be a moneymaker. But we don't like it because it doesn't make sense, but at least you have done it. I mean, it's not that you can just skip an extractable study, but you have done it, you have the knowledge, you already know what are the extractables of your device, and that gives you also the data and the knowledge to go forward to a simulation study. In a simulation study, the number of extractables is much lower, let's say between 50 and 200. It's still a lot, but also manageable to some extent. And then hopefully, when you're doing your toxicological risk evaluation, end up with no toxic extractables and you can release your product to the market if all your other testings were okay as well. Another case study is that if you're working with permanent implants, a knee, a pacemaker, a stent, how can you simulate a device that is in direct contact with the patient? How to simulate the contact with blood, maybe the lymph solution or fat and so on. That's becoming difficult. And that is why we are sometimes faced, look, we have blood contacting devices. We use 40% ethanol, 50% ethanol, something like that, which is mostly accepted by, for instance, US FDA. But you can also be creative. There are cases in saying, look, we are having an implant in the bladder. Contact solution is urine. You can try to work with artificial urine to do your simulation study. So the temperature can be simulated. You don't need to use 50 degrees C by definition. You can work with 37 degrees C as well. So thinking outside the box can really help. And yeah, that's really it. Whatever the patient contact is, you can try to find good solutions for that. Another example is, yeah, it's again a bag. I have something with bags today, it seems. It's meant for short-term storage and also flow-through of blood and blood products. Yeah, you can do an extractable study on it, and I guarantee a little bit the same result as for the enteral feeding bag. A huge lot of extractable compounds, and a toxicologist will say, these extractables at these concentrations, it's not safe for patients. Well, okay, let's work on the simulating conditions. Let's work on the simulating solvents, but we can also work on the quantifications, because we have done a first study and we know which chemicals are causing problems for a toxicologist to make his judgments or her judgments. So we can work on the quantification of the analytics. For instance, we can include calibration curves, we can work on the responses and investigating everything to improve the quantification and saying, okay, before we knew that a concentration was, let's say, 1 milligram in a device. Now we can say, okay, it's not 1 milligram, but it's 0.75 milligram, or it's 1.2. Having better quantifications helps the toxicologist in overcoming potential tricky situations. Improve the identifications is also possible. As mentioned before, the med device industry is very innovative. The universe of the extractable compounds is always growing. Unidentified extractables is a reality that we face. Every lab faces it. And trying to identify those as much as possible is key. Sometimes you need very good equipment. If you have your screening techniques, you have sometimes your unidentified compounds, and at Nelson Labs, we can take this to the next level with next level equipment as well. Basically, yeah, if you're talking about the specifications of the instruments, it's for instance a GC QTOF machine or an LC Q Executive or a machine that is really capable to measure the mass of the molecules very, very, very accurate. They have fragmentation possibilities that other machines don't have. And what you are basically doing is you're making a jigsaw puzzle. You all have fragments with very accurate masses. Based on that accurate mass, you can make an elemental formula like 2 carbons, 4 hydrogens, and an oxygen, for instance, based on basically the very accurate mass. Then you can try to build structures, and that's just a textbook example on how to build structures based on fragments, and you come up with an identification. At the top left of the screen, once you have that structure, we can purchase an analytical reference standard of that compound, and we can confirm our theoretical jigsaw puzzle identification. And then that's how you can upgrade your data so that the toxicologist does not have to assess something, yeah, I don't know what it is, then the toxicologist can assess a true chemical with known structure, and that's of course much better science. So, just to make a short conclusion, prepare your chemical characterizations very carefully. Don't skip your theoretical exercise. prior to do the actual extractables testing in the lab. Design your study, taking into account all that information, that knowledge. Sometimes you know up front that you will be faced with certain problems and challenges. Don't ignore that, but anticipate on that. You can anticipate with your prior knowledge from yourself, knowledge or experience from the lab that you're involving, to already anticipate on simulation studies. After which you can still work on improving the identification or the quantification strategies. But most importantly, is don't panic. Don't panic. Be smart. Use your knowledge, and together we figure it out. And that's it. I would like to thank you, and if you have any questions, I'm happy to take them.